Okay, so when we started chapter 3, the title of it was Biological Molecules. What does that mean? Molecules that are within living organisms, so within life, okay? I also, at the very beginning, I had a slide, and it said, this is what else you might hear biological molecules referred to as. What are some other names for biological molecules? Organic molecules.
Inorganic.
Not inorganic. Macromolecules. Organic macromolecules, or biomolecules. All the exact same thing. It just depends on who's speaking about them. So if it says something about a macromolecule in a question, you already know what a macromolecule is. Okay? Now, so we went through that first, and then we talked about the importance of carbon. And we also mentioned this in chapter 2, but carbon is an element that we find in all living things. And we talked about that organic means what? It has carbon. That's all organic means, is that it has carbon. Okay? So if it's organic, it has carbon. But if it has carbon, does it have to be organic? No. And I need to make sure that you have the ability to distinguish between the two. So if it's organic, it has carbon. But just because it has carbon does not mean that it's organic. Okay? So we discussed that. Then we talked about hydrocarbons. Do you recall what hydrocarbons are?
Carbon.
Just carbon and hydrogen together. That's it. So you have carbons bonded to hydrogens. You have now hydrocarbons. And we discussed that they could be quite simple or big and complex. So they could be linear, they could be in a ring structure, but hydrocarbons just means it has hydrogen and carbon. Okay? Then I talked about immediately after that, like a compound that you can attach to these hydrocarbons. Do you recall what I'm referencing? Something that you attach to these hydrocarbons. Functional groups. And what do these functional groups do? Tell that molecule, this is the way we act, this is what we do. By definition, the functional group is the reactive portion of that molecule. So it's the part that's going to actually carry out the chemical reaction. Okay? So we talked about the functional groups. Then we went on to talking about, and I redrew the metabolism diagram, and we added some terms. We talked about taking water away and adding water. Yes, very good. So when you take water away, what is that doing? We call that dehydration. It's bringing molecules together. It's anabolic, it's building up, it's creating bonds when you take water away or dehydrate. Okay, and we put different words that you might see substituted in there. And then when you add water, what are you doing? You're breaking things apart, and we call that hydrolysis. Or you could even reference it as hydrolysis. Either way, it's the same. It's just where you put your emphasis. Okay? So we talked about adding water to break things down, and that was catabolic. And we talked about taking water away, which was dehydration, condensation, synthesis reactions, which were anabolic. So we discussed that. Then we went into covering the actual biomolecules. And there are only 4 biomolecules on Earth. What are they? Carbohydrates, lipids, proteins, and nucleic acids. Okay, and on whatever day we were together, we did carbs and lipids. And I showed you a chart, and I said, this is kind of how I know what you need to know for the test. And on this chart, I had the name of the biomolecule, then I had a column for AKA, or also known as. Then I had what elements does it have. Okay? Then I had the function, what's the monomer, polymer, and then the type of bond that you will find within those biomolecules, that class of biomolecules. So we started with carbs. And carbs are also known as sugar. Okay? What elements will I find in carbohydrates? Carbon, hydrogen, and oxygen. Fantastic. Okay? And what's the function of a carbohydrate? Energy storage. Structure. There was energy and structure for carbohydrates. Energy and structure. And what was the monomer? Monosaccharide. And when you hear the term monomer, what does that make you think? Just one. So the smallest a carbohydrate can be is just one sugar. And the monomer for a carbohydrate is a monosaccharide, which literally translates to one sugar. Okay? Then we talked about the fact that what if you had one sugar and another sugar, and I wanted to bring them together, what type of reaction would that be? That's the type of bond, but what's the type of reaction? Dehydration, synthesis, condensation. This is bringing it together. And I am, yes, creating a glycosidic bond. That is 100% correct. Okay? Did I add water or take water away? Take water away. Very, very good. And if I want to continue to add 5 more sugars, how many more waters do I need to remove? 5, because remember it's a 1-to-1 ratio. Okay, so that was our monomer. Then we also put in our bond there, a glycosidic. What were the examples of polymers, which we called polysaccharides, which translates to many sugars? Starch. If it's a starch, where did it come from? Plants. Non-negotiable. We use it for energy. What's another polysaccharide? Glycogen, which comes from animals. You and I have glycogen. This is where we store our energy. Okay? Cellulose. Is that structure or energy? Structure. And where will I find cellulose? In the cell walls of plants, which make plants completely indigestible to animals, which we talked about them and how we leach them for minerals and vitamins in some water, but we use them really just to clean out, and they help us poop. What was the 4th carbohydrate polysaccharide we talked about? Chitin. Chitin. And chitin, I told you, when you hear this term, not only is it structural, but you're referencing one of 2 things. So what are the 2 things that we would be referencing that have chitin? Fungi. Fungi and arthropods, the exoskeletons of arthropods, like, for example, a grasshopper, cricket, daddy longleg, anything that has a crunchiness to it, a crab, a lobster, shrimp, all of them are considered arthropods. And their exoskeleton, which is their skeleton on the outside, is composed of the carbohydrate chitin, which is structural. So that was carbs. Then from carbs we went to lipids, and I said lipids are also known as fats. Fantastic. And what elements will I find in lipids? Carbon, hydrogen, and oxygen, which are the exact same ones I'll find in carbohydrates. Yes. And then I asked, what's the function of lipids? Energy storage, insulation, and protection. These are basic functions for lipids. I asked, what's the monomer? The one that you and I are going to use for this course will be a triglyceride. So the fact that it says tri means it has 3. It had 3 fats on it. It had a glycerol molecule and 3 fats. What did I have to do to join each of those fats to that glycerol molecule? Dehydration. How many times did I have to dehydrate? 3. I created 3 bonds. I needed to dehydrate 3 times. What was the name of the bond I created there? Ester. Ester was the type of bond there. Okay, so we had our monomers. We talked about our ester bond. What about polymers for lipids? Neutral lipids, which are like your saturated, unsaturated, so solids, waxes, oils. Those are all neutral lipids. Then you said phospholipids, which we find in the cell membranes of all living organisms. And steroids, which we use as hormones or chemical messengers. And that's where we left off when we left on the last time we were together. So we are now going into the 3rd biomolecule, which is proteins. 3rd biomolecule, which is proteins. Does anybody know what protein is fancy for? Just protein. Protein. Just protein. Protein is fancy for protein. As far as the elements found in protein, carbon, hydrogen, oxygen, and which one did we add here? Nitrogen. Now the functions of protein. I have here, proteins perform many functions. Literally, there are thousands of functions. However, we're not going to list thousands of functions. So I kind of put just a few, but these are ones that I think are important for you to list because we will use them within this course. So structure, transport, communication, and then I capitalized and emphasized enzymes because we will have a full chapter on enzymes. So I'm trying to pre-teach that here. So as far as functions for proteins, structure, transport, communication, and enzymes. The monomer for a protein, and again, monomer means the one, is an amino acid. The monomer for a protein is an amino acid. And there are only 20 amino acids on Earth, and I'm going to come back to this point in just a moment. But it's not like there's a bunch of them. There are only 20 to choose from. Okay? And then I have here this very last statement. Amino acids, which are the monomer, are held together by peptide bonds to form proteins, which are called polypeptides. So I gave you the name of the bond you will find in proteins. So if I have two amino acids and I want to join them together, what type of reaction? What type of reaction will help me join? Dehydration or condensation. Okay? What's the name of the bond I have between two amino acids? A peptide bond. If it was a carbohydrate, it would be called a glycosidic bond. If it was a lipid, it would be called a ester bond or an ester bond. Yes. Okay? Fantastic. Then I have this little structural model for an amino acid. And I have this here for a couple of reasons, and you'll see that I'll tie it in just a moment. But when I know I have a carbohydrate, I either have a hexagon or a pentagon. Like that tells me you're dealing with the sugar. And when I have a fat, it's kind of long and it looks kind of like a flag because of all the greasiness to it. Well, you can't see greasiness in a structural model, but we'll get there. Okay, but here when I have this amino acid, I have functional group, hydrogen, and then I have an R. Whenever I'm looking at things and trying to figure out what it is I'm working with, I immediately know I'm dealing with a protein when I see this. If I see that R group, I'm like, oh, that's protein. Like it's not a carbohydrate, and it's not a lipid because it has nitrogen. What do carbohydrates and lipids only have? Carbon, hydrogen, and oxygen. I have a nitrogen, so I know it's not a carb or a lipid. So that either leaves me with a protein or a nucleic acid. I have this R here. That tells me I'm dealing with a protein. We are going to end up calling that R a variable side chain or an R group. A variable side chain or an R group. But I just wanted you to see how I can visually decipher what I'm dealing with. Because you're going to have to do the same thing. Now here's the chart straight out of this current textbook. I don't expect you to write anything for this, but what I did want to show you is that I mentioned that proteins have many functions. Support, increase the rate of a reaction, so on. And what can you see also changes as their function changes? Their shape. And where do you think I'm going with this? Shape determines function. Yes, the shape will determine the function. And if you leave here, this class, in life, and you never again understand that shape determines function, I have failed you. Like, that's what it is. The shape of everything determines what it's capable of doing. Okay? This is continuing with that same chart, again, straight out of your text. You have the function and the shape, and of course it describes what's going on there. You're not going to have to know each of these. My point was to show you there's multiple functions and there's different shapes because the shape determines the function. Yes. So now we're still on monomers. And you're not necessarily adding anything to the chart. This is information that you just need to be familiar with for your test. So again, an amino acid is the building block of a protein, so it's the monomer. But here I have this same structural model, and here I have defined what each thing is, because I know you'll need to know that. So I tell you here, we have an amino functional group right here. That's where the nitrogen is. This is an amino group. Then it tells you here we have a carboxyl. And then it tells you here we just have a hydrogen hanging out by itself. And then we have a variable side chain or an R group. Here it specifically says variable side chain or R group. So you have both written there for reference as to what we could call that R. A variable side chain or an R group. This part will always be the same. The only thing that will change will be what's here. Do you remember how many amino acids I told you there were? 20. Do you know how many different proteins there are? Thousands, hundreds of thousands. Real quick, anything that you can physically put your hands on is made of protein. Your skin is protein. Your hair is protein. Your organs are protein. They may have other biomolecules as well, but the actual structure of that is protein in nature. How do I know, just by looking at you, that all your proteins don't have the same shape, that they're all different shapes? How do I know that by looking at you? Yeah, because you have a different shape. Like your hair is a different shape from your skin. Because if all those proteins had the same shape, you would just look like one big same shape. Like there would be nothing distinguishing the characteristics of you. Okay? So we have 20 amino acids and we have hundreds of thousands of proteins, potentially. So how, if we only have 20 amino acids, do we have so many options for proteins? Because amino acids are what make proteins, and there's only 20. But we can make thousands of proteins. They have the side chains, there's only 20. They do have different shapes, but how do I get that? How many letters do we have in our alphabet? We have 26 letters in our alphabet. How many words do we have in our language? And we're adding, true or false. So when you say a lot, we're talking like 100, 1,000. There's hundreds of thousands of words in our language. And you could venture to even say millions. So if I only have 26 letters, how did I get that many words? The combination of the letters. If I change the order of the combination, I change the word. If I change the order of the amino acids, I change the protein. Yes. If I change the order, I can come up with a different protein. So yeah, I only have 20 options to use, but I could put them in all different orders and create an infinite number of combinations for proteins. So we only have 20 amino acids. 20 amino acids, but we have thousands of proteins that are made from those 20 amino acids. It mentions here that those R groups, which are those variable side chains, they differ in size, shape, and reactivity. They differ in size, shape, and reactivity. And you never are going to have to memorize them for this class. But you will need to be able to recognize them. We'll use them in this chapter, and we'll use them in a future chapter as well. Here is the chart of amino acids, and I chose this chart because it gives me a little bit of information that ties me to a concept you've already learned. I have 20 amino acids. These all have the same base. Do you see how it's the same? What's the only thing that changes? The variable side chain, which is this part here, and as a result of this changing, it changes the structure. Yes, you're correct. So Every single one of them has the exact same hydrocarbon base, but we change their side chain. I can group them as a result of their side chains. So those first 9 have a nonpolar side chain. As an educated student, when you see it's got a nonpolar side chain, what does that tell you about those amino acids? They don't mix with water. They are hydrophobic. So you knew what nonpolar was, and as a result, you can deduce what that molecule is capable of doing or not doing. It's not going to mix with water. This group here of 6 has a polar side chain. What does that tell you? They're hydrophilic, that they easily associate with water. You know that just because you know what the words are. You don't know specifically what serine does, but you know here, based on this chart, that it's polar, which means it will work with water. Here, where it says the electrically charged side chains, these have various impacts, and I don't expect you to know them already, but I did want to show you this because I wanted to make sure that you understand that when we talked about polar and nonpolar, we weren't just talking about fats. Like, it applies to everything, yes, okay? And this, for some of you, is overkill. For others, it's just reinforcing the content. It makes you more comfortable. The first thing I want us to address is when I look here, how do I know that I'm dealing with a protein? That R, that side chain, here. Because I can look at this and see, oh, structural model, ooh. But once I see this, I know I'm dealing with a protein. Okay? Once I see that variable side chain. There's also something on here that tells me I'm not dealing with a carb or a lipid. It's got nitrogen in it. It has nitrogen, so I know it's not a carb or a lipid. Okay? So, I do now know that I'm dealing with the protein. I have my side chain, and I have that N. And here I have 2 amino acids, which are the building blocks, the monomers. And when I want to bring these 2 together, what do I need to do? I need to dehydrate them. Take the water away. So I will remove an H2O. The type of reaction is called a condensation or a dehydration reaction. Okay, synthesis, anabolic, we talked about all of those terms. Once I remove that water, I have it here on the side. These 2 are now joined, and what's the name of the bond that holds them together? A peptide. It's clearly labeled, okay, but I want to keep walking you through this. If I want this to be broken, this peptide bond to be broken, what can I do? I can add water. Hydrolysis. Okay, and when I add water, it will separate that peptide bond and it will break it down. It's a catabolic reaction. Case in point, why you should write those words on the back of your Scantron. You're thinking the right answers, but some of you are saying, and it's happened about 4 times now, and of course I'm not going to be like, you're wrong, but you're saying the wrong answer, but you're following along with what I'm doing. So I know you know what's going on, you're just choosing the wrong word. If you have that diagram there, it's right there. You already know, oh, adding water, removing water, you know what's happening. Okay, and so you know what answers to pick based on that. I'm just trying to give you a tip for preparing for the test. Okay, on this diagram here, this did come out of the notes that were posted on eCampus. So I'm going to talk about shape, but before I get way into shape, I wanted to take a minute and discuss these terms. These were not on eCampus specifically defined, but I saw some area of gray. And if you're a student who is going to analyze and second guess your answers, these are words I want to make sure I address. First of all, an amino acid by itself, when you have just an amino acid, that's a monomer. So we're straight on that. But when you have a group of amino acids, that is less than 50, so you have amino acid, amino acid, amino acid, amino acid, amino acid. What are the name of these bonds here? Peptide bonds. When I have less than 50 amino acids, I don't call this a protein yet. If I have less than 50 amino acids, I refer to it as a peptide. This becomes confusing for students because peptide is also the name of the bond. Okay, so I'm clearly addressing that. The definition of a peptide when we're talking about a protein means that it has less than 50 amino acids. But it's also the name of the bond that is formed between those amino acids. It officially becomes a polypeptide by classification, meaning many peptide bonds, when there are over 50 amino acids dehydrated together. Okay? So if there are 12 amino acids, what is the classification? Peptide. What if there are 63? Polypeptide. What if there's one? An amino acid. Your question.
So when you say 50 amino acids, that means like 50 of the same one, not
They can,
like the 20 that you said that there's.
of the 20, there's a combination of 50 in a row.
Okay.
It could be, it's not likely going to be all the same amino acid, but it could be any combination of those 20 whenever you talk about a protein. Any combination. Yes, ma'am, that's a great question. Okay, so these were terms that I wanted to address and say specifically straightforward. This is what it's referencing so that you're not confused. Okay? A polypeptide is not officially a protein until it has multiple polypeptides attached to it. So a protein is many polypeptides. A protein is many polypeptides, usually 3 or 4. Okay? Now, let's address this structure here. Go up. Okay, on this structure, initially, when we're just doing amino acid, amino acid, amino acid, adding each of these, what structure is this? It's linear. Does anyone know what the term primary means? First. So the primary structure, the first structure for a protein, is linear. So as you're initially adding the amino acids, it's linear. Okay? As you're initially adding the amino acids, it's linear. And this chord isn't going to be the best for a demonstration purpose, but it does carry out basically what I'm trying to address. So as I continue to add amino acids, so I had that linear structure, if my proteins get really big, can my body create more room for them? No. So as I continue to add amino acids, that protein will start to coil. It'll curl into its secondary structure. What does secondary mean? Second. So you can see that second, it starts to coil. We actually call that a helix. We call that a helix. You may also hear it called a sheet, like a pleated sheet. So it says that there. It's either an alpha helix or a beta pleated sheet. But ultimately, the protein, the amino acid chain is getting longer, but I don't have any more room. So it starts to coil or twist to fit in that same space. And then what happens as I continue to twist and twist and coil? What eventually starts to occur? It'll start wrapping and kinking. It'll form like this mass tangle. Let me give you an example, and this will help some of you understand this. If you had an older phone before it was cordless, and some of you have them on your desks. Some of you are like, I have no idea what you're talking about, and that is okay. No judgment passed on you, but you're not old. Okay, so in my lifetime, cordless phones were invented. Like I remember when we always had just corded phones, and then all of a sudden we had a phone that you didn't have a cord, and you're like, what? Like cell phones came out in my life. Okay. But when I lived in Oklahoma, my parents had a phone in the kitchen, because you usually had just one phone, maybe two, hooked to the landline, right? One in the kitchen or the main family room, and then one in like your parents' room that had the cord. And our first phone, the cord was kind of short. So when I would talk, you had to like stand there next to it, and you're in the kitchen, everybody could hear your conversation. And it really didn't matter, because I was, only 5 or 6 then. But you had to stay in a certain spot. And I remember when we all of a sudden got a longer cord. And if you know what I'm talking about, you know what I'm talking about. Like, sometimes they could be like 12 or 15 feet. And now I didn't have to stay in the kitchen. Like, I could walk to the living room and talk, and that was cool, because I could, like, walk around, right? Or you could go hide and talk, and nobody could see you. They could see the cord, but they couldn't see you. So the cord itself was coiled like this alpha helix, okay? And as I stretched it, it kind of got thin. But what happened as I walked to put the phone back? It started to coil and tangle. That's what our tertiary structure looks like. And what does tertiary mean? Third. So after I get past that alpha helix and I continue to twist and coil, it starts to tangle. Bless you. It starts to tangle. And that creates our tertiary structure, which is our third structure, which just happens to also be three-dimensional. So this is when it officially becomes three-dimensional. So tertiary, three-dimensional. Okay? This, by definition, is a polypeptide. Okay? My tertiary structure is three-dimensional. It's a polypeptide. What does quaternary mean? 4. This is the 4th and final level. And it's showing you that this polypeptide right here is here, but I have also included 3 others. So I have 4 polypeptides here. This right here is a protein. This right here is a protein. It's got 4 polypeptides. This is a protein. So I went from my linear primary structure to my secondary structure, which could be a helix or a sheet. Then as it continued to kink and coil, it formed a tangle, a three-dimensional tangle. That's the tertiary structure. And then when we go from tertiary to quaternary, so from 3rd to 4th, I take not just 1, but I add 3 more, and now I officially have my protein. So these are all labeled as polypeptides. You can't see those 2. So I have 4 polypeptides together, now I have my protein. The first day that we lectured, I said, a preschooler can recreate this shape, but is this shape random? No. It has a specific shape because this protein has a specific function. This has a specific shape because this protein has a specific function. Questions right now? So this is telling you shape determines function. It's just reiterating that. And again, it's not random. Just an example. And then, no surprise, I say it once again. Protein shape and function. The shape of a protein helps to determine its function. You know this. All right? It says proteins that lose their shape. So if they lose their shape, what's about to happen? They're going to lose their function. We actually have a term for that when we talk about proteins. Proteins that lose their shape, we refer to as being denatured. If you see any derivative of this, so denature, denatured, denaturing, denaturation, it's all the same. Okay? It's all the same. To denature means to change the shape of that protein. What are some things that could cause a protein to change its shape? Heat, sterilization, acids, UV radiation, irradiation, high salt content. These are all things that could cause a protein to lose its shape. Let me give you an example that applies to you. First of all, I told you anything tangible in your body has protein in it, okay? How many of you know what your brain feels like right now? Like if you were to touch your brain, you feel like it would be squishy. It is squishy. Your brain, at this moment, has the consistency if you had a tub of butter and you left it out overnight. So it's not that solid like it came out of the refrigerator. It's that like warm room temperature butter. What's going to happen when your finger touches that room temperature butter? It goes right through. Literally, if you were to put your finger on your brain, it would go right through. That's what the consistency of your brain is. When we do autopsies right now, when we take your brains out, we cut your skull cap off, so we remove your skull. We can't just go in and grab your brain, and we can't start cutting at your brain. We can't. We have to pull it out with the meninges, which is the covering on it, out, and you have to either freeze it or put it in formalin to preserve it. Because if we were to start cutting or messing with it then, it would just melt away. We couldn't do it. That's your brain right now. Mostly lipids, but of course there's protein there as well. Okay? Now, what happens when you run a fever? What's the purpose of running a fever? Okay, it helps you fight an infection. How does running a fever fight an infection? It makes you sweat, so it makes your internal environment hot. And what does heat do? It denatures. It will start to denature the bacteria, or try to. That's its goal, to denature whatever it is that's infecting you. Okay, there's also some other aspects of running a fever, but the main one I want to get at right now is the heat. But your body says, wait, you're getting really, really hot. And when you get really, really hot, what does your body start to do? After you've had the fever for a while, you're hot. What do people with fevers start to do afterwards? They start to shiver. Why do they start to shiver? They get cold. And then they cover up and they warm up, and then guess what happens? They get hot. And then we get too hot, their body says, you're too hot, and now I need you to. What is your body trying to maintain? Homeostasis. Because if we need the heat to kill what's ever in there, right? But too much heat will kill us. So we need to go back and forth and regulate that temperature. Okay? So if it gets too hot, we tell our body it's too hot and it cools off. Guess where we lose heat from the fastest? Our head and our feet. Okay? Case in point, when you get really, really hot at night under the covers, tell me what you do. You could kick out one foot. And then, no, there's no. And then after you lay there and you get cold with that 1 foot out, what do you do? Just slip it back in. Like you don't flip the whole cover off unless you're dramatic, and some of you may be dramatic, but you just stick your foot out. And if you're hot and you're a female and your hair is covering your neck, what do? You put it up because that's where we can release heat, okay? So that's where we release heat naturally. So when we're hot, we're cold, you and I are able to regulate our body temperature. So that keeps our own proteins from denaturing. Guess what age group of humans is unable to regulate their body temperature? Infants. Infants. When a child is first born, what do they do at the hospital? They wrap it and cap it, and sometimes socks. Yes. Okay, because they're trying to help that baby stay warm. And when that baby gets too hot, what does he do? Cries. And then you unwrap him and he cools off. And then he gets cold and then he cries. And then you wrap him. Because he or she cannot regulate their body temperature. So when that child begins to run a fever, why is it such a big deal? He can't cool himself down. When we're maintaining homeostasis and we have a problem, we fix it. What type of feedback is that? Negative feedback. Negative feedback. You have a problem, we fix it. You and I, we're hot, then we're cold. We're hot, then we're cold. That's negative feedback. A baby gets hot, and it continues to get hot. What's eventually going to happen to the proteins in that baby? They're going to denature, and that's going to lead to irreversible damage. Okay? So when we talk about proteins, of course, it's really difficult to take something so abstract and make it make sense to you. But when you think about why is it a big deal when a baby runs a fever, Well, because they can't cool themselves down, and those proteins will denature and cause damage that cannot be reversed. Okay? The same type of damage can be caused by having too much salt or using UV rays. This can all cause all of those, that denaturing. But that's just an example that could help you remember what it is that I was trying to get you to understand. Any questions on denaturing? Okay, last biomolecule. Yes, ma'am.
I'm sorry. I don't think, what was the polymer for proteins a
Polypeptide. Yes, ma'am. The polymer for proteins is a polypeptide. Okay, the last biomolecule is a nucleic acid. What is nucleic acid fancy for? DNA, RNA, and I'm going to throw another one in there because it's going to come up later, so we're pre-teaching. ATP. Energy, fantastic. So AKA DNA, RNA, ATP. Now we aren't going to discuss ATP today, but at least I know that we've referenced it and we'll come back to it. So DNA, RNA, and ATP. The elements found in nucleic acid, I have my carbon, hydrogen, oxygen. I also have nitrogen, but in addition, phosphorus. So I've added another element, so another level of complexity. Another level of complexity. So what's the function? What's the function of a nucleic acid? A message, okay? So if it's a message, what is it carrying? If you're a message, you're carrying information. DNA and RNA, these are nucleic acids are information. So they store information. And remember the first day of class, first lecture that we had. I said everybody knows what DNA looks like, but they don't know what DNA does. DNA stores information, but information to do what? Makes proteins. Fantastic. It stores the information to make proteins. Stores the information to make proteins. That's what we use it for. To make proteins. How do you know that I have the DNA for blue eyes? Because the proteins I make my eyes blue. You can see that. Do you understand what I'm saying? Like, so you know that that's specific. How do I know that you don't have the protein for blue eyes? Or the DNA for blue eyes? You don't have blue eyes. Okay? We'll learn to start thinking in that way. So that's the function. Now the monomer, which again means just one, for a nucleic acid is called a nucleotide. It's called a nucleotide, but right beneath it, I have three things listed because a nucleotide has 3 parts, which I know you have to know. A nucleotide has 3 parts. It has a nitrogenous base, and I want to point something out on this nitrogenous base. You may see it written like this, nitrogenous base. You might see it written like this, nitrogen base. You might see it say nitrogen-containing base, but they all mean the same thing. They all mean the same thing. They all mean the same thing. So we have the nitrogen-containing base, then we have a 5-carbon sugar, then we have a phosphate. So we have sugar, phosphate, and a nitrogen base. The sugar, phosphate, and the nitrogen base are the 3 parts of a nucleotide. And a nucleotide is the monomer for a nucleic acid. Here at the very bottom it says nucleotides, which are monomers, are held together by phosphodiester bonds. Phosphodiester bonds to form nucleic acids, which are the polymers. So the monomer is a nucleotide, and the polymer is a nucleic acid. What's the name of the bond? Phosphodiester. If I say ester bonds, you know I'm talking about lipids. If I say glycosidic bonds, you know I'm talking about carbohydrates. Fantastic. Phosphodiester bonds are for nucleic acids. Everyone good so far? So here are two of those nucleic acids, DNA and RNA. Again, most people can readily recognize DNA as being a double-stranded molecule. RNA is just a single strand. Here it has the second point of difference for these two nucleic acids is that in DNA, the sugar is called deoxyribose, and in RNA, it's called ribose. But watch, physically, you can see this. Because ribose has an O and an H, an OH group here. What do you think deoxy means? To remove oxygen. So instead of having an OH, I just have an H. So it's ribose if we just take an oxygen off. So this is ribose, deoxyribose. Okay? The next characteristic that distinguishes the two are the nitrogen bases used. And see here, it doesn't even say nitrogen or nitrogenous. It just says bases. In DNA, there are 4 bases that could be used: thymine, cytosine, adenine, and guanine. And usually, we will reference them just by the first letter. Okay? Usually it will say like A, T, C, G. For RNA, the bases that are used are the same except for we have uracil, which is U, instead of thymine, which is T. So RNA has a UCG. Adenine, uracil, cytosine, and guanine. So these are the major distinguishing characteristics. This is also a bonus question on one of your tests, so I wanted to make sure that I addressed it so that you're not like, she never discussed that. Here you have it. Okay? What's the monomer for a nucleic acid? A nucleotide. A nucleotide. And this rendition right here is basically what I would draw if I had to draw you a nucleotide. I would have my sugar, my phosphate, and my nitrogen base. And I would just keep it simple like this because that's how I think. So this is just a basic diagram of a nucleotide. Whereas, this is what it actually looks like. Which is why I don't try to draw it. But I have this right here. What biomolecule is this? How do you know that's a carbohydrate? It's a pentagon. Okay, and it's got CH and O. This is a carbohydrate. It's a sugar. It's clearly called deoxyribose sugar. It's a deoxyribose sugar. But is a nucleic acid a carbohydrate? No. It's just showing you that, well, first of all, I just wanted you to identify it because I want you to be a beast. But it's showing you that these are all part of what we collectively call a nucleotide, which is the monomer for a nucleic acid. Okay, so it is clearly labeled as a sugar, and we know that all sugars are carbohydrates. So here's my sugar, here are my phosphate groups, and my nitrogen-containing bases. What it actually looks like. Okay, here. This is the nitrogen bases. Okay? Nitrogen bases are divided into 2 classes. The ones that have just 1 ring and the ones that have 2 rings. If they have 1 ring, we call them a pyrimidine. If they have 2 rings, we call them a purine. This is one of those things that I memorized. So it took me a minute to stare at it and realize how I was going to memorize it. But then I looked here, and I said TCU. TCU are pyrimidines. A and G are purines. So TCU is pyrimidines, and the Aggies are purines. That's how I memorized it, and that's literally how I still remember that. It's just memorization. Is this going to change in the next textbook? No. No, it's not. Okay, again, to be a pyrimidine literally just means you have one single ring there. And to be a purine, it means you have two. Questions here? Here is just a diagram showing you, there's a couple of things I wanted to show you here. First of all, what's the type of bond I'll find in nucleic acids? Phosphodiester. Phosphodiester bond, and those are located here along the sides. Okay? Phosphodiester bonds. And DNA has two strands, and these two strands are held together by these bonds. Do you remember what these bonds are? They're hydrogen bonds. How do you know they're hydrogen bonds? Dot, 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 dot. Are these strong or weak? And I didn't say are weak, I meant to say or weak. These are weak bonds. DNA easily separates, and we'll talk about that in a future chapter. But I want you to recognize this stuff, because the more you can recognize, the better you're going to do. Okay, so again, phosphodiester bonds are located on the actual joining of the nucleotides, but hydrogen bonds put the bases together, the nitrogen bases together. What bases do we expect to see together? A and T will always be together. C and G will always be together. A and T will always be together. C and G will always be together. A and T will always be together. C and G will always be together. If you're dealing with DNA. I had a freshman biology at the high school student who is studying, and she said there's apples in the tree and the cars in the garage. So she remembered it that way. I'm telling you, biology is memorization. If you can figure out things, it'll apply to all your higher sciences as well. But RNA is different because RNA does not have a T. So instead of being AT, it's a U. Okay, let me tell you where this is going to come into play. On your test, you'll have a strand of letters that looks like this. It'll say, identify this biomolecule. You will have to say that it is, what is it actually? How do you know this is DNA? T. This is DNA. So you'll have to be able to identify it visually, and this would be then RNA because it has U's. Okay? That's the first thing. Second thing is, a question may say, you have 100 nucleotides. And of those nucleotides, 32 are adenine. How many guanines do you have? So if you have 32 A's, what do you already know? I already have 32 T's. Because whatever there is A's, there has to be that many T's. So these two together make 64. That means whatever I have here, these two have to collectively equal 36. So I would divide 36 by 2 and get 18. So the answer would be there would be 18 guanines. It will not be very difficult calculations. That you'll have the back of your answer document. And don't forget, you can also bring a small scientific calculator, basic, not your phone and not your TIs. But you can use those because for some people this is a lot of calculating, I get it. Okay? So make sure you have those tools. But that's what a question may look like, just so that you're ready and you know what to expect. Okay? This last slide references the fact that these bases are complementary. What does the term complementary mean? Complementary. It goes together. Like when you have peanut butter, there would be jelly. It's a complement. Or I had a student say honey, peanut butter, whatever, they're complementary. If you have a tennis racket, you would expect to have not a tennis racket and a football. Those aren't complements. So, if you have an A, you would expect to have a T if you're talking about DNA. And if you have a G, you would expect to see a C. Okay, so you can see that occurring here, where those complements are together. So this is showing you, this is actually a pre-teach option 2 for DNA replication, but we won't get there today. This is the end of chapter 3. What questions do you have? Everybody okay?